This video aims to discuss the main points, results and conclusions of the question, how do endocytic defects contribute to the progression of cancer, and what possibilities does understanding these defects offer for development of novel therapies? The first part of this question is regarding problems in either the receptors or the machinery of endocytosis, and this involves a number of different signalling pathways. One of the receptors discussed is called ERB-B2, and the results of studies done on this receptor show that it has a link to breast cancer. The ERB-B2 receptor has also been found to activate the RAS-MAPK signalling pathway when it is mutated. RAS can also activate PI3K, the signalling pathway of which is involved in clathrin-independent endocytosis. The PIK3CA gene encodes the P110 alpha subunit of PI3K, and is an oncogene that when mutated can lead to cancer. PTEN is another protein in the RAS-PI3K signalling pathway that inhibits AKT, which is normally activated by RAS. Studies have shown this by looking at the loss of PTEN and how it affects cell proliferation, and the results have shown proliferation to increase, meaning that PTEN inhibits the RAS pathway and that activation of RAS is important in oncogenesis. The P85 alpha subunit of PI3K has been shown to stimulate the activity of RAB-GTPases, some of which have a role in endocytosis. In particular, RAB5A, which is involved in membrane fusion, can be mutated and was found to promote cell proliferation. This was discovered to be mediated through the FAK signalling pathway, which can also be activated by P53. It was revealed that overexpression of FAK leads to increased cell growth and tumour progression. P53 is a key regulator of endocytosis and has been found in mutated forms that activate MET signalling. MET endocytosis plays an important role in tumorigenesis, and it was discovered that an increase in the endocytosis led to increased metastasis. The activation of MET signalling can in turn activate SRC signalling. This has also been found to cause tumour progression through the stimulation of the FAK signalling pathway. The FAK signalling pathway can also be activated through integrin. Mutations in P53 increase the recycling of integrin and facilitate its binding to RCP, which is an oncogene and therefore has the ability to become a proto-oncogene and induce cancer. Dynamins are proteins that aid in the pinching off of vesicles from the cell membrane, and both the overexpression and the reduced expression have been found to be involved in some forms of cancer. The second part of the original question is in regard to how the knowledge of these signalling pathways can help us to develop new treatments for cancer. There has already been research into therapies that target the ERB-B2 receptor, and another receptor that has potential to be a target is VEGF. VEGF endocytosis regulates the process of angiogenesis, without which tumours would not be able to survive. HSP70 is another potential target, as it was found to be a regulator of clathrin-independent endocytosis and was also found to be overexpressed in some tumours. In conclusion, many of the defects in endocytosis are found to be overexpression of many of the signalling molecules, as well as some of the receptors. The signalling pathways all activate each other and ultimately lead to an increase in cell growth and migration, which are main characteristics of tumours. Most of the identified defects are not known in great depth and are so not at the stage of being used as targets for therapeutics. However, there are some that have been researched and successful treatments produced, and hopefully the research will continue in the future to develop more novel therapies.